we are very glad and fortunate to have with us Dr. Praveen Kumar Mamidala. Sir will be delivering a lecture on science, technology, and innovation. Dr. Praveen Mamidala is currently serving as Joint Secretary, Higher Education, TSWREIS, Government of Telangana since August 2018. Prior to this, Dr. Praveen worked as Associate Professor and Head in the Department of Biotechnology, Telangana University, Nizamabad. He has 21 years of teaching and research experience, which include teaching in Osmania University and autonomous institutions. Dr. Praveen achieved distinction with university rank in MSc Botany, Kakatiya University, Warangal, and MTech Biotechnology, JNTU, Hyderabad, and completed postgraduate diploma in Patents Law from prestigious Law University, Nalsar, Hyderabad. Dr. Praveen received several national and international fellowships, international training course at Biological Research Center, CZ, Hungary, under ITC Fellowship, the World Academy of Sciences, Italy Chinese Academy of Sciences, China Postgraduate Fellowship in 2004, TWAS-CS Postdoctoral Fellowship in 2008, UGC Postdoctoral Fellowship in 2006 and 2007, Dr. D.S. Kothari Postdoctoral Fellowship in 2009, Ohio Agricultural Research Development Postdoctoral Fellowship, tenable at the Ohio State University for a period of 3 years, 2010 to 13. Awarded International Bilateral Exchange Program to visit Israel and Visiting Scientist Program 2016 by Indian Science Academy, New Delhi. During his doctoral and postdoctoral work at China, Dr. Praveen developed transgenic tomatoes that showed resistance to high salt concentrations. During postdoctoral work at University of Hyderabad under guidance of Professor Appara Podile, Dr. Praveen has developed nanoparticles for post-harvest management in tomato. Dr. Praveen developed huge genomic resources for ash species, emerald ash borer, bedbug, potato leaf hopper, red palm weevil, using high throughput sequencing methods during his third postdoctoral work at United States of America. Till date, Dr. Praveen has published 52 research articles in high impact factor journals with total citations of 879 with H index 18. In the recent past, Dr. Praveen received grants from Department of Science and Technology, New Delhi, worth rupees 48 lakhs and several other grants for organizing seminars and conferences. Besides the major projects, Dr. Praveen Laboratory is funded with SERB Young Scientist Postdoctoral Fellowships and DST Women Scientist Fellowships. Dr. Praveen has presented several invited talks, more than 52 in national and international conferences. Dr. Praveen is a reviewer of several peer-reviewed scientific journals, like PLOS ONE, Insect Molecular Biology, Journal of Insect Physiology, Plant Cell Tissue and Organ Culture, Plant Molecular Biology Reporter, to name a few. Dr. Praveen served in various administrative positions at Telangana University, Nizamabad, such as Controller of Examinations, Coordinator UGC and Non-UGC, Director SC/ST Cell, Member Secretary IBSC, Member Secretary of Institutional Ethical Committee. We welcome you, sir, and request you to take over.
Thank you very much, ma'am, for spending lots of time on my intro.
Thank you,
think sir. I am audible, right?
Yes, sir, you're audible.
Okay. Very good evening, one and all. It's really, let me thank at the outset to Dr. Shailaja and American Corner for inviting me to this 4-day faculty development program. It's an apt time on effective teaching of STEM courses online. I will make it a little change, like science, innovation, technology in a sequence. Instead of STI, let me make SIT. I will be focusing more of my time on only 2 slides. I prepared slides with my research experience and also relevant to the society, whatever we are doing, the science. Because people, if you ask anyone STEM, they are confused. They feel that STEM is something like engineering, or STEM is something like motivating student towards, say, science field or technology field or something like that. They are in a confusion. So before I go into my topic, I am a blessed teacher, I can say, because generally a teacher journey goes like this. If you are teaching for a school, you'll be promoted as a junior lecturer, and from there you'll be promoted as a degree lecturer. If luck permits, you may also enter into university. But my journey is in the reverse way. I worked as a postdoc and worked with a postdoctoral team and came back to teaching for post-graduation students in universities. And later I came into degree college, a reverse way. And then I was told to go and do some experiments in the intermediate colleges. And now I am doing experiments in the schools. It was really a completely reverse order in my journey. So why I'm saying all this is with my experience, what I understood is if they're given the opportunity, I want to work in a school, particularly in the elementary section. I want to give up all this university teaching or postdoctoral work. I will tell you the reason why. When you come to STEM, when someone asks you about STEM, which is a really an interdisciplinary approach, do you think we have that approach in our colleges or schools? I think no. If you ask someone about STEM, they start doing STEM activity from 8th class or sometimes from 10th or sometimes from intermediate, no wonder. Or also from degree, they will say that take STEM course. It's like a jargon like now. Take STEM course, go abroad. You will get OPT if you are in the STEM, if you are in the science field, particularly the American corner guys. If you ask any American embassy guy, they say that take science related courses because you'll get 4 years OPT and you can stay happily, you'll get a job. But it is something and a great approach where a student start doing lots of experiments at the age of 4, 5, 6, 7, 8, 9, at a very young age. He has lots of curiosity. He has lots of logic. They have lots of creativity. Unfortunately, we will kill that all these factors, and we simply say no, do this, do that, and get into BPC or get into MPC. You will make compartmentalize. The entire life goes compartmentalization, and they will end up with nothing. They may get a degree, I'm sure, but not the skill, whatever they are thinking. So what I want to say is getting a STEM education, of course, can lead to definitely into STEM careers. But what exactly we have to do, what exactly a student has to take up a particular science course, whether he is eligible to take a science course or not, whether he has a logic, he has a memory, he has kind of ideations he will do, or he has a multitasking. If you ask a science teacher, because I'm a biology teacher, I'm a botanist. Basically, I'm a botanist turned into entomologist, later into this very fashion word omics, because I was in USA, I did omics. I will share my experience also about the omics later. So if you ask any teacher why that biologist, what is that biology student must have, whether he or she must have a logic or curiosity or out of box thinking or some experiential learning or say engaging or higher order thinking, all these things. Otherwise, whether they have to do project work, why they have to do project work for the STEM, why particularly for the STEM students. In the recent past, if I'm not wrong, the UGC has released a order saying that every degree college must do a project work. Then if you don't do project work when you are in the schooling, suddenly you are told to do project work in degree. What is that you will do when you don't know? Why I'm telling this is, my dear friends, when some PhD students come to me, like who want to enroll with my lab, they ask me always a question: sir, I want to join in my lab. I want to join in your lab. Then I'll ask, why you want to join in my lab? Then say, sir, I want to do good research. Then I ask, what is that you want to do? Sir, whatever the question you give, I will do. Whatever the project you give, I'll do. But they never come up with their own question. This is unfortunate. This is not in my lab perhaps, but go across, even in the central universities also, majority of students, they don't know what is the question, what is the research question. There we got stuck. So this all happening because we are not awareing our students on this STEM approach or making, you know, interested towards STEM. So If you can go to next slide, ma'am, if someone can help me. Of course, this is very simple. Everyone knows this, but I want to focus on one or two words. Whenever you say science, science is something special where you need to have some great observations and experiment. When you have an observation, when you do or when you plan an experiment, then from that or with some idea or with some a new method or whatever, then you will get some output. Unfortunately, is we are stuck at observation. I'm not saying that everyone, but majority are in the India, students particularly, also teachers, they simply close eyes, and it's kind of a rote learning and uh, complete rote learning, vomiting on paper. So you will go and just write, fill the paper, and you'll get the award degree. The degree getting 70%, 80% has now become very routine. Earlier days, getting 60%, 30 years back, it's a tough job. But now you see in the science, they get 95 percentile, 95 percentage or whatever. So they do not do any observation and they do not plan any experiment. When you don't have this, you are gone. You cannot do any innovations also, and you cannot get any technology also or whatever. So as with my experience, even if you ask now a MSc postgraduate or a teacher about patent, you ask a word patent, they are confused. Sir, patent is not my cup of tea. It is something law, some other course. I'm not sure, sir. I studied somewhere in my master's as a one unit, and in also in the bachelor's one of the question, but I really don't know. I don't know. If you ask an MBA student, entrepreneurship is a Greek word to them. If entrepreneurship is a Greek word to a biologist, to a MBA student, definitely it will be a unknown word to the biologist. So when the biologists do not know about entrepreneurship, when a biologist do not know what exactly his or her future is, definitely you should not talk about anything about technology. You should not talk anything about innovation. When you don't, when you do not know, understand the societal problem, because we see science and society as separate domains. We never see them clubbed together. When you do not know the societal problems, your science cannot be applied. When you do not know what is exact problem and how to resolve the problem, and when you do not know how to do an experimentation, I think we cannot come up with any technology. That is the reason why I will show you a next slide. I think from there you'll understand. This is a simple criteria, very, very simple, very simple. Everyone, I think they might be knowing, or some might be new to this. If you want to have a patent, like what are the criteria? If you ask any teacher, they say very simple, three criteria they should have. One is important is novelty. Novelty is nothing but the invention, whatever you are making, must create a new knowledge, like a product or a process. Like it should not be anticipated by the document or granted patent or say published patent or non-patent literature. Like it should not be in the public domain, and it must be different from what is already known. Simpler terms, novelty. It should be novel, ekdam novel. That's all. And number two is like the step, the inventive step. In the patent, in the legal terms, we say it's non-obviousness, but in the simpler terms, you can say, what is that? It's something which is not expected by the person skilled in that art. It should be something like a different. It should be like completely non-obvious. The best example is a PCR, Carrie Mullis. I request all the teachers who join this webinar, go to Google and try to download it's very freely available, the patent of PCR, maybe in the June 1987, which was filed, and it almost took 3 years to issue a patent on the PCR. You will get a very good drafted patent. From that, you will understand how much tough it is to draft a patent, and also what is the innovation behind the PCR machine, the PCR steps or whatever. And finally, very important is, this is very, very important. That's what I written, patent on papers, patent and perish. So I have many of my friends, they say I have 2 patents, I have 3 patents, I have 10 patents, just like that. Nowadays, if you go and ask any student, I don't say Usmania University or some Telangana University or some other university. I don't want to blame universities. They say they have 50 papers in the PhD, or they say they have 20 papers in the PhD. I don't know how they publish within 3 years or 4 years or 5 years max of the PhD time, publishing 50 or 40 or 30 papers, which is an impossible. So all these are in the kind of predatory journals. The same is happening with the patents also. They claim that they have patents, but whether that patent has an industrial applicability or not, that is an important one, my dear friends. So these industrial application patents are granted to ensure that the inventor can exploit their invention freely without the fear of competition. This is one of the important things. And invention should be used or manufactured in the form of a product or a process. So this is all about these three important criteria. But where India stands in all these things, just go to next slide. I think someone can help me to go to next slide. There you will get the annual index. Can anyone help me go into next slide, ma'am? Okay. Or can you give me access? I can take over.
Gayatri or Ravi, are you there?
Yeah, thank you very much. So this is an intellectual properties index. This is released annually by the US Chamber of Commerce Global Innovation Policy Center. So what it will do is it evaluates intellectual property rights across 53 global economies on the basis of patent and commercialization of IP assets, copyright policies, and ratification of several international treaties, all this stuff. You can see India somewhere on the left side. So they have you can see luckily this is a 2021 ka hai, but if you go just 3 years back, it was on the complete end. And slowly it is shooting up thanks to a little bit science and technology improvement in India since last few years, when all CSR labs told that we don't want to give funding to CSR labs, and until and unless you come up with patents, don't focus on papers. Then they started working now to come up with some out-of-box ideas, go for some collaborations, go create some STA hubs, science technology hubs, and understand the local indigenous issues, problems, and then intervene and try to produce some really applicable, industrial applicable patents. Now little bit we are doing best, not worse though. What I want to say is, when you don't have science with you, you cannot get a patent. I'm in the first slide only again. When I go back to my first slide, is observation and experiment. When you don't observe, you're gone. I want to share 2 of my projects with all the teachers because it looks very simple, but I got around 1 crore rupees from those of 1 crore. People say always that what is DST, what is DBT, what is UGC. They don't give money to us. They give only to those people who have some connections with DST, DBT, or who are familiar with some professors in the screening committee. No, it's all, I am sure, it's all with your research question. I will share one of my experience in my next slide. So as I told you that observation and experiment is a science, and ideation. This is an important part where this must happens in the classroom. This must happens in the classroom, and also this must connect to the society. Do you think our teachers go into ground? When I say ground, some agricultural fields or into the urban space or rural space, they never go. They will hardly read textbooks, or they'll hardly use our Google. Google has become a great book to us. Whatever you doubt, just type it on the Google and get some meaning or get some understanding of that. So ideations will only happen, the brainstorming will only happen when you take your students and go into the field. So, and once these 2 are done, the technology comes. I just made a smartwatch here. Lots of technologies now. I don't know where it's going because of this IT. I'm sure by next 10 years, we will be seeing lots of products with IT, particularly the data, all data. Like we have some tons, some mets. I don't know how to measure that much data we have. That's why now data analytics is ruling. The AI or machine learning or NLP or whatever. All these are coming just because of this data. So, but still, even if you want to do that data, or even if you want to do that coding or whatever, you should go back again, see what the society exactly needs. That's a very important one. I want to tell you on 2 slides, very important slides. I think you'll understand what is the importance of science, because when you don't understand importance of science and simply jumping into lots of tools, actually I thought of sharing you what are the STEM online tools, all this, but I thought instead of giving that, I can share you the PPT. There's no need to, because it's available on the net, I can simply copy, and I don't want to do like that. I thought I should explain why this is important. Okay, next slide. See the beautiful flower. This is a gerbera. I think everyone knows about this. No one should say I don't know, because wherever you go, you will find this flower. Okay, but ornamental flower, different kinds of colors you have. So one of my postdoc, he established a small greenhouse where he started doing business with the gerbera. And one day he came to me and said that with a very sadness, sir, there is no good market. I thought I should earn lots of money, but there's no good market, sir. Top on this, I don't know how to control the pest problem. There's a huge pest problem, sir. I'm spending all of my money on pesticides. And he observed something in the field, and one day again he came back, sir, I don't know. Some flowers are okay, some flowers are not that much healthy, and the market will not take such damaged flowers. Then I asked him, his name is Sai, he's a very good research scholar from University of Hyderabad. I asked her, Sai, why don't you go and observe which are the flowers which are damaged more, or which are the flowers which are not damaged? Then one day he observed for 4 or 5 days, and he quickly came back to me, sir, all the white one or looks very healthy, sir, but this yellow is completely damaged, and the pink, unfortunately or unfortunately, in some cases white has demand, in some cases the yellow flower has demand. Then I told him, Sai, simply try to bring 4 pots and say put it in separate containers or separate room, and see that all these are healthy first, white and pink, red, all these. First select them, and then take some thrips, okay, because thrips will damage like anything, and put colonies, equal number of colonies on each flower, and leave it for 1 or 2 days and just go and observe. And this guy did that experiment, very simple experiment, doesn't need any money. He has a ready-made pots and everything is there, and he did the experiment in 4 days, that's all. And what we observed is there is no much problem on the white, but there's a huge infestation, like huge multiplication of thrips on the colored flowers, but not on the white. Meaning what? Very simply, if you ask anyone, they say white might be resistant and yellow might be sensitive. That is the obvious answer of what we expect from a layman also. So, but what makes it resistant, what makes it susceptible or resistant is the science behind we need to understand. So what we did is go to next slide. Yeah, so yeah, so this is another interesting, I still remember a great research paper written by Dan Hems. Dan Hems is a great evolutionary scientist working in Ohio State University, and he published a paper, very good paper, that the dilemma of plants in the annual review of biology, whether plants grow or defend, what is the trade-off mechanism. For example, when a plant is subjected to huge stress, it may be a biotic stress or it may be abiotic stress. It will plant to grow and die quickly, or just go on defending and waste its all metabolic resources, or without making its next progeny. This is a very interesting paper. If you have some time, please read this paper. It was published in 1992, The Dilemma of Plants to Grow or Defend. This is an excellent paper, my dear friends. It's openly available in the Google. You can just type it by Dan Helms. So what I want to tell is, on the left side, whatever you're seeing, these are thrips, and the right side is the salt stress. So very interesting. What we understood is, what we analyzed is, or what we hypothesized is, the white flower is resistant to the thrip, okay, and the yellow flower is very kind of susceptible or easily attacked, right? Then I asked, okay, if this is resistant, this may be sensitive to the salt. And if this is sensitive in the case of thrip, it may be resistant to the salt. I think I'm not confusing you guys, but once again, I want to say the white flower is resistant to the, say, thrip. Definitely, it should balance its all internal system, and somewhere it will become sensitive. In the same way, if yellow is sensitive, it may be resistant to the salt. Based on these simple observations, I think these are very, very simple. Of course, we have done some mortality test, we have done some antioxidant and some other preliminary kind of test to prove that our hypothesis is correct. It cost not more than 10,000 rupees, but we got 1 crore rupees, my dear friends. 1 crore only on these questions. First question is to compare transcriptomics of white with yellow, and to compare the metabolomics of white and yellow, and to see the salinity stress in the across the 3 cultivars in the gerbera. See, only one observation. One observation will take you to lots of research. So such a kind of observation is needed. When you come to this society, we have lots of problems, but we never try to address those problems, or we never look into those problems. And we simply say that science has nothing and we can't do anything because India is a place where there is no good lab, there is no good tools. Only if you go to USA, you can do. If you are in India, you can't do anything because India is very bad. It is wrong, my dear friends. No, you can do wonders only when you have that observation skill and kind of developing an experiment for your proving that your observation is correct and that your hypothesis is correct. So yeah, we'll go to the next slide. If yeah, this is what I told, that we took the 3 different cultivars, white and pink and yellow, different flowers. Of course, the variety, the names are different here. We say Latara, Basic, and Faith variety, and we observe like after 3 days, post 7 days, and it's a percentage of mortality. This shows that we did some, as per statistical analysis, replicates we have done, all as per a very good design experiment, and later we found that yes, the white is resistant, and this followed many biochemical experiments to prove that okay, this pattern is correct. And then we entered into the omics. I don't want to say about omics story because everyone knows about omics, what happens in the transcriptomics, what happens in the metabolomics or proteomics. It's a snapshot at a given point. What are the compounds, or what are the proteins, or what are the transcripts that really express and probable candidates for the resistance. That's what is the simpler terms of omics approach. We fish it out. Work is going on. It is not yet published as of now. And now from here, I will take you into a different world. It's really very interesting because, as I told you, I'm a botanist and turned into an entomologist, not by choice, actually. It is like for the what we call as for food, or you can say for getting some money, extra money when I'm working as a postdoc. I went to Ohio State University to work on some plant genomics or plant transcriptomics. When I got some extra time, I used to work. When you work extra in America, that's very great. You'll get some money, maybe $1,000 per month, or sometimes $2,000 if you're lucky, if your boss is good. So next slide, please. Yeah, this is what I told you about omics approach. I'm not going into details. Next slide. Yeah. So you can see on the top, good night, sleep tight, don't let the bed bugs bite. I think the age group of, say, the college students, they do not know much problem about the bed bug, but say 50 years or people who born somewhere in at least up to 80s, they might experience bed bugs. I'm not sure. Particularly in the rural spaces, they might experience it. In the urban spaces in India in late 90s, they experienced it when we stopped DDT, and then all these bed bugs, like they wash it off and they migrated to Europe, and from Europe somehow they entered into the USA. This is what the scientists predict, but again, there is no evidence at all. People simply say, man, all these bed bugs came from India. But again, there is no clear evidence for this. It's prediction. So bed bugs really made a nuisance. It is a kind of a nuisance pest, I can say, particularly in the Europe and USA up to 2015. Now it's not a big nuisance, but before that, it is a highly nuisance pest. So it's a huge loss to hotel industry. It is huge loss to places where the students reside in the dorms. And also, it's not a great threat to the human health, not a great, but though still people used to fear about the bedbug bites. So that's one day, just we are sitting in a lab and we were thinking how to get funding for my lab, because your lab always run with your own funding. Unlike India, once you get a job, you are very happy. I always say with my friends, once you're a government job or once you're in the federal or state or whatever, you simply sit and you'll get monthly your salary. Simply sitting monthly getting formula. That always I'll say. Even you do work, you teach, or you do not do any research, you'll get money. But in US, it's not like that. Definitely, you have to fund your lab on your own. So it was like everyone is like thinking what to do, what is this bedbug doing, how to stop this bedbug nuisance. Everyone is thinking. On one fine day, when we sat together, three brains, okay, me and one of my postdoc friend, and other is my PI, my supervisor. When we thought, when we are working on a plant system, resistance versus a susceptible plant, we thought, okay, these are the bed bugs which are exposed to the insecticides or which are exposed to the various pesticides. Can't we compare the genome, the transcriptome, preferably simply because during those days, it was all in 2010, exactly 10 years back. This paper got published in 2011. Can't we compare the bedbug transcriptome with the pesticide exposed strain with that of the strain which never exposed it? Then again, we got a doubt where from we should get this never a strain, a bedbug which we never exposed. And then again, we thought how to get this. And we thought, why not check with some museums? Luckily, we found a museum in New York. Thanks to that Adam, a great guy who's a passionate, animal biologist, entomologist, not biologist. So he was culturing the bedbugs since last 40 years. And he has a bedbug, a particular strain, which never exposed to any pesticide. He was continuously growing in the lab, continuously culturing in the lab, all with all the love, because there are some passionate people, they want to do this museum, like maybe animal museum or insect museum, something like that. So we asked them, can you give us a few bed bugs? Then he told, okay. Then he asked us why. Then we told, yeah, we want to compare and see the patterns or find out something. And very simple experiment, my dear friend. People talk much about omics. Omics is nothing a labor can do. Only thing is you should have idea. Only thing you should have a tricky mind to fish out the genes. That's all. Otherwise, it's a very simple work. Just isolate RNA from the two different strains and give it to a company. Even you no need to prepare library. Of course, way back in 2010, we prepared libraries on our own because it was so costly, and it took cost time, not more than 1 month though. But we prepared libraries, and we compared the entire transcriptome by DE-seq analysis, like differential expression sequence analysis using Illumina platform. At one go, we got thousands of genes where there is lots of upregulation and downregulation. And next slide, you can see that pattern. Yeah, this is what, see, when you have an idea that has got some impact in the society, this happens. A very small discovery, but it touched BBC, the Lantern, the Wall Street Journal. Everyone started praising us. Wow, they reveal bedbug genome. Wow, there's a clue what's happening with bedbug. That's a noise we made across the US borders. Next slide. So this is a very simple pictorial diagram I was telling you, taking this and with the help of Illumina platform and followed by a little analysis, we found that, okay, these are the genes which are highly expressed. These may be candidate genes, and then we validated using QRT-PCR. Next slide. So what we did is we tried seeing the tissue-specific transcriptome. We have few tissues, important tissues to always look into for such experiments. One is cuticle, the outer covering layer of the insect. Other is the Malpighian tubules, because lots of detoxification happens in the Malpighian tubules, midgut and fat body. So we checked this gene, which was expressed high in the in silico analysis, and then we found that, okay, this is some candidate to do. This is something interesting candidate to do. So then to prove this, again, we have to see whether if you do silence it, and then only you can confirm. Within no time, big labs from Harvard, big labs from Baylor College of Medicine, everyone jumped into this project. Oh, some candidate gene is there. So of course, I have not silenced this gene, but people from Baylor College of Medicine, they silenced, and later they developed lots of products from this research output. Next slide. Yeah, this is the different, if you take bedbugs, we have 3 important stages. When they are born, newly born, we say early, and after a little time, we say late, and then adult. And we found that there is a differential expression across the developmental stages. And then we did some docking studies, why it has become resistant, what is the reason, what might be the reason, because you spray any pesticide or any insecticide, it will not die. The only reason is whatever we thought the cytochrome P450, a candidate gene, when we did some homology modeling, when we did molecular docking studies, all the it can block all the insecticides or whatever you are using. Next slide. It will show very clearly. You can see that picture. Next slide. Yeah, this is the picture. Of course, I'm not mentioning the DDT, deltamethrin, and the various pyrethroids. All this, this is used to detoxify whatever the compounds which they take inside. So from this, we thought, yes, we need to silence this, but you need to understand its full gene. And we quickly jumped into RNAi. Next slide. Yeah, we try to see, again, no one did RNAi until we did for the first time. So we need to inject this dsRNA somewhere very cleverly. and we try to inject safely so that there is no mortality with your injection. And we found that we tried very cleverly. We tried with Malpighian tubules. I just made funny to make a bedbug kidney, but it was with Malpighian tubules. And we observe. See, when an insect takes huge blood meal, okay, what happens is it cannot move. Okay, if you see mosquito, when it bites you like this, and when it takes huge meal, it takes some time to fly. So within that time, it has to excrete. So it means that its Malpighian tubules are fully functional. So we got the idea: can we block some of the very important genes like potassium invert rectifier, we say Curie genes, so that it will die. So our expectation was good. It died. So the RNA was working. The moment, as I told you, Jack Alderman, I guess his name is, from Baylor College, they started working on the cytochrome P450. Anyways, whatever the hypothesis, what we did is correct. And what I want to say from all of these observations or experiences is, when you closely look into, for example, if you are working on an insect, you should feel like an insect. When you're working on a plant system, you should feel like a gerbera flower, or you should feel or think what's it. And again, that particular content or subject or whatever you are dealing with must help your society. It's not that if I do in bedbug or someone doing on some other insect just like that. No. If you, that's why I was telling, I was asking my PhD students every time, please come with some question here. Come with some questions. Come with that to your own question, and with a very good hypothesis driven, very good objective. So there you can do impact your society. This is not happening, my dear friends. So let's go to next slide, my dear friends. So yeah, the significance of the project, whatever it is the first time we made some genetic makeup, and we understood like what are the various metabolizing pathways, identifying candidate genes, and which made basis for developing our interference, and the novel bedbug genes, the novel, what I said, that will really represent, what best control targets, and how it got adapted to the naive ecosystem, or how it really become established in various places. That's what we understood from this project. Only very simple idea. Can we compare a bedbug transcriptome and with that of the a susceptible one? That is the only idea. Earlier also the same. See which is susceptible, which is resistant. Very simple observation. Next slide. Yeah, so what I always feel is online tools, all those tools now has become very buzz. Just now before I'm coming into this lecture, I watched excellent lecture by ma'am. She was telling more on MOOCs and some other various online resources. Yes, but perhaps the best resource, the best lab on the earth planet is our brain. That's what I feel, really. Honestly, the true laboratory is the mind, where, behind illusions, we uncover the laws of truth. Fire, what a great invention. Wheel, steam engine, awesome, right? Electricity, microwave, apps. I can't say apps now. I don't know in future what apps will do. Apps definitely will control us in coming days. They will tell us whether to eat or not. They will tell us whether to walk or not. Everything is now in future. Apps definitely will control the human beings, I'm sure. So it's all, the mental models or the laboratory. So we need to tell our students to understand the society first, to understand the problems first. Do you think we don't have problems? We have lots of problems. Unfortunately, it's the Western world look into our problems and start working on that. We in India, we never look into our problems. We try to look into the problems of the Western world. Some diseases, I don't want to name those diseases because you much know better than me. But instead of looking outer side of India, come back and see our spaces, particularly rural spaces, what they are suffering like. Is they are suffering with water? Is they are suffering with some other domestic helps, or what is that they are suffering, and what is the idea you have to save them, or to to make them lives better, as not as our lives, but at least to live as human beings, which is not happening across the India. If you go to rural spaces, when I became a joint secretary for higher education, I started going across the state. Now I have complete geography of the Telangana state. We always say that we work with marginalized people. That is a word we use, unfortunate. Till date also, our brochure on social welfare says that we work with the marginalized communities. Why they were marginalized? Like, why, if you see the, I understood complete Telangana geography. If you go to North Telangana, Adilabad, if you go to South Telangana, Alampur, if you go to the Khammam, Bhadradri, Kottagudem, these are the areas. I think in this FDP program, at least some people are there from Telangana. If you see this map, what you understand is all this map is dominated by the poorest of the poor. The core may you don't have the poor people. Core matlab, it may be Warangal, it may be Hyderabad, it may be Karimnagar. But if you go to the periphery, if you go to the borders, really they are marginalized. So what our science really can transform their lives is a very important, my dear friend. This is what I thought of asking or telling you. Next slide. So as I was telling you that the mental models, online labs, I really, if you can give me some time, actually, I'm really sorry to say this. When Shailaja asked me, sir, you have to give a lecture, then I started working on making lots of online tools for the STEM. But I realized that instead of saying all those STEM online tools, this is the one, like Go Labs, we have several online platforms, of course. Instead of explaining that, if I make this opportunity to make my teachers or these FDP participants, whoever, to motivate them and now go at least, onto the ground and understand what are the problems, I thought that should be the best one. But I really, maybe in a couple of days, I will share all the list with Dr. Shailaja, so that she can share to all these participants who are there. And again, coming back, can we do some scientific experiments? Answer is yes, we can do. And very importantly is every student, do you have some question? And very importantly, I did the experiment since last 3 years, my dear friends. Everyone knows that flipped class. I think if I say flipped class, they say, yes, sir, I know flipped class. But teacher do not keep in practice. This is happening across the Western space, but not in India at all. No, we never make a flipped class reading. We never make a student-centric approach. If you can make your students, or if you can make your community to read at home and come back to class and start working on problem solving. So it doesn't happen in the classroom spaces. It may happen in college like St. Francis, I'm not sure. But for in the university, no one, majority, 90% of teachers, they never think on these problem solving sessions during the classroom spaces. So can we make such inquiry-based learning or flipped class? Recently, when we have this national education policy, new policy, I written on my own I submitted to NEP that HECI is one of the things happening in coming days, like Higher Education Commission of India, all the regulatory bodies or the big shots, all they are coming under one umbrella. But if these HECI and NCRT and the school boards, they don't come together and work together. Just now in my intro, I was telling you that UGC is introducing project in degree. So up to degree, you don't know what is project, and suddenly you ask to do project, what they will do? 25 years back, I remember, project means do some experiment and make spiral binding and submit to a teacher and get some marks, 25 marks out of 25. And I think now it may be 30 marks or 50 marks, let's say 100 marks. And again, sell off that project book to juniors or give to juniors. That's what's happening in the projects. But whether we are really doing this or not, this is the question we have to think. So when you go back to our colleges, I know it's very tough game because I worked in the private college also. Like you people, there are some private college teachers. I always feel private college teachers are the best teachers because, they work very hard. When you come into government space, my sincere request is don't join in government space. Don't join into any government college, a degree college or a university. You are gone. Your passion, your seriousness, commitment, everything is gone. So, but you don't have much freedom in the private college. Your management may listen to you or may not listen to you, but ask them, request them, sir, we want to change this, the complete the way of learning, teaching, or assessment, or whatever, pedagogy, whatever. And if you do some changes and connect to the society, if you don't connect the society, if you go on talking something, that's end up with nothing. That's what happening since last 2 decades. What I observed is many of the cases, many of the FDPs, many of the places, people take very seriously when they are listening or when they're attending. But the moment they go back to the colleges or because of some other reasons, I do not know, they do not put them into practice. My sincere request is please try to start change or transforming lives of these marginalized communities. At one point, we should keep full stop. How long we should say that India has got marginalized people, India has got poverty, India has got this, India has got that problem. At some point, I thought we should stop saying such words and see that we have a very good, a very good system for education and also for careers. So yeah, I told that I will take around 45 minutes because I want to have some interaction with the teachers. If you permit, I will take some questions, or otherwise I will say bye. Over to Shalja ma'am.
Yeah, thank you, sir, for this, such kind of, the lecture, which has really motivated all of us as teachers. We find our role is to motivate our own students to, look around, carry out a good observation, come out with something which is interesting. But the problem lies in how much, as you said, there are a lot of other things that are involved. And then, even if a student has a question as a research problem and she wants to do something, lack of time is something which I think is the major, because the semester system, everything has to be wound up in that particular limited time.
Correct.
And you're running behind your assessments, your internals, your main end exams, your practical exams. By the time it ends, the next semester begins. Lack of time is one major aspect, I think.
Correct.
When we work as teachers, we find so many students, coming forward,
Yeah.
wanting to do good research activities. At least, if not at the even at the undergraduate level, I feel
Yeah.
I actually get many students in microbiology who are really interested and motivated towards research. And even
Correct.
at PG level, we have good students who really want to work hard. But time is one aspect, I think, is which is withholding them back. But as you said, yeah, role of a teacher is to motivate the students, to carry out, to look into the problems of societal needs. We need to, push. But how exactly we can go about doing it is something which, many of them lack. Can you enlighten us on that?
Yeah, a good question. And what I want to say is once we understand the subject or the topic which we are dealing with the students, once we have the depth of that, once students get a depth of that, then there will be having a kind of brainstorming, and from that brainstorming we will get lots of clues what to do or how to go about. But only thing is, as you rightly said, it is not happening in the colleges because it is fully packed. It is like tightly packed. The entire semester we are running, just like that. So syllabus complete karna, we have to complete the syllabus within no time, and then do assessment, do some internal marks, and get prepared for final examinations. Again, prepare for PG exams. So this is our focus. But in the classroom space only, when we are taking up a topic, let us give them a question every time to think on, a question. Otherwise, they never come up with thinking at all. They always last 20 years I can see, because people like your age group, or I think I do not know your age group, but people of the 45 years, I think they know the seriousness of this, understanding the science. But later, it has become just like kind of what you call as just learn whatever it is, copy and put it on paper. So they never understand in the science. But if you can create interest in science, I think we can bring
Maybe as a
something.
teacher, we need to, put up a discussion platform,
Yeah,
class,
discussion.
wherein, let them come out with new ideas and try
Exactly.
to, capture those ideas and motivate
Correct.
them and tell them
Correct.
that, okay, you've got a very good idea and you can go ahead and doing this and how to plan about. Maybe we can give a direction.
Direction. True. True. Yeah. Yeah. Yeah.
Yeah.
Yeah. I
And,
agree.
we do a lot of things like, we ask the students to come up with their ideas for
Oh, really?
anti. Yeah, we do that in the class. We tell
Okay.
them to get their ideas and showcase their ideas in front of the
That's
faculty.
great.
And
That's
then we try to motivate them. This is one of the best ideas that we have heard about. And then, such kind of sessions, maybe they would help. And research projects, yes, definitely one of the important aspect that every science student should have, whether it is life sciences or physical sciences. All of them should get into project work, because that designing your own experiment is another aspect, I think, which is very interesting, where they only come up with their own experimental design and how to go about doing it. Unless and until they do it, they will not, taking information from somebody and implementing it is not a good way of doing it. It's you're designing your own, you have an idea, you design your own experiment, you
correct.
try to work it out, you find faults with that, and you come up with newer ideas. I think that kind of motivation will really help them. So maybe as teachers, we have to do this kind of a role. And true, as you said, looking at your both ideas and projects that you have carried out, it's really motivating and nice, sir. Thank you so much.
Thank you, ma'am. Thank you very much.
Any other questions from the participants? You can keep it in the chat box. Now I request Dr. Rosalind, Department of Microbiology, St. Francis College, to give the vote of thanks for the session. And participants, the feedback form is provided in the chat box, so please fill it up to receive your certificates.
1 minute, Sunita. I think Malti has mentioned that it's one of the motivational talk that sir has given. Thank you so much.
Okay. Thank
Just
you,
one
ma'am.
comment on the
Thank you very
chat.
much.
Really, it was very interesting and nice, sir. We would love to, listen to many more such kind of innovations, which would really motivate everybody to go into research. Thank you.
Thank you, ma'am. Thank you very much.
Yeah, Sunita, you can continue.
Yes, ma'am. Over to Rosalind.
On behalf of American Corner Hyderabad, Departments of Microbiology, Biochemistry, and Biotechnology, St. Francis College for Women, I deem it a great honor to thank our esteemed speaker, Dr. Praveen Kumar Mamidala Garu, Joint Secretary, Higher Education, TSWREIS, Hyderabad, Telangana, for an enlightening session. Thank you, sir, for motivating us with your talk on science, innovation, and technology. We're grateful to you for sharing your research and teaching experience. Thank you, sir, for highlighting the importance of patenting, patentability criteria, project, and research ideas. Your talk is truly an inspiration for us to move towards innovation and be effective teachers. Thank you, sir.
Thank you, ma'am. Thank you very much. Thanks to all the panelists, and special thanks to Dr. Shailaja, ma'am, and also to the American Corner. Thank you very much. Namaste.
Thank you, sir.
Thank you, sir.
Bye.
I would also like to extend my sincere thanks to American Corner Hyderabad for their association in conducting this international workshop. I take this privilege to thank Mr. David Moyer, Public Affairs Officer, U.S. Consulate General, for gracing the inaugural session and sharing the activities by American Corner and importance of STEM education. I take this time to thank Dr. Joe Christopher, Public Affairs Section, U.S. Consulate General, Hyderabad. I take this honor to thank Dr. Sister Sandra Horta, Principal, St. Francis College for Women, for the welcome address and encouraging words. I express my sincere thanks to Dr. Sister Shirley, Vice Principal, and Dean Academics, Dr. Mallika Shetty, for their support. I'm extremely thankful to our speakers, Dr. Anil Kumar Challa, Professor D. Mamata, and Dr. M. Praveen Kumar for sharing their expertise. I'm grateful to Dr. M. Shailaja Raj ma'am, convener and head Department of Microbiology, for conceptualizing the workshop and sharing the themes and importance of the workshop. I also thank the co-conveners, Dr. P. Kanchana, Head Department of Biochemistry, and Ms. Shauni Nivedita, Head Department of Biotechnology, for their efforts. I also thank Dr. Anita Thomas and Ms. D. Sunita, faculty from Department of Microbiology, for moderating today's program. My sincere thanks goes to Mr. Ravi Kumar, Coordinator, American Corner, for his support and guidance. I thank Dr. Gayatri and Dr. Annapurna for their support in smooth conduct of today's sessions. I would like to thank Mr. Ram for the technical support and also thank each and every participant without who this workshop wouldn't have been successful.